The title says, Link between long telomeres and long life is a tall tale, study finds. Now, when I saw that title, I thought, oh, brother, another supposed debunk of the hypothesis that I advanced in the very late 90s on the relationship between telomeres, which are repetitive genetic sequences at the ends of eukaryotic chromosomes. We are eukaryotes. Eukaryotic chromosomes are linear, they're not circles. And telomeres are repetitive sequences. They're not genes. They are just sequences of DNA that repeat at the ends of chromosomes. I'll get more into what they're about shortly. But in any case, when I saw this, I thought, okay, this is going to be another one of these articles where they claim, without pointing to my hypothesis directly, but they debunk the idea that telomeres mediate a balance between tumor suppression and tissue repair, the balance between them creating increased longevity. That's what I thought when I saw this title. And then I started reading the article, and holy moly, that is not what this article says.
The New England Journal of Medicine article or the New York Times article?
The New York Times article. I will get to the New England Journal of Medicine article that Gina Colada, the reporter who did the New York Times piece, is reporting on in a second. But contrary to the implication of that title, and I will say I think Gina Colada has a lot to answer for in this article. It's very poorly done, frankly, but she's probably not responsible for the title. The title is probably...
Well, and you've got a headline and a subheadline, and the subheadline looks to tell a slightly different story.
This subheadline does tell a slightly different tale. It says, the longer a person's telomeres, researchers found, the greater the risk of cancer and other disorders, challenging a popular hypothesis about the chromosomal roots of vitality. Now, I find that subheading also very misleading because the point is the telomeres are at the root of vitality, but it is not more telomere better. It's a balance, much as I argued back in the late 90s, and then with my co-author Debbie Cizik published a paper ultimately explaining the hypothesis and the evidence surrounding it. So if you will scroll down to that first paragraph, Zach. So this again is Gina Colada, one of the New York Times science journalists. She says, the story, as often happens in science, sounded so appealing. Cells have a molecular clock that determines how long they live. If you can just stop the clock, cells can live indefinitely. And the same should go for people who are, after all, made of cells. Stop the cell clock and you can remain youthful.
Well, I mean, but that paragraph is directly responsive not to your hypothesis, not to what you have published in the Journal of Experimental Gerontology back in 2002, but to the sort of pop medicine version of that, on which basis presumably tens, if not hundreds of millions of dollars of grants have been given.
Indeed.
Right? Like that is what she is reporting on there is truly something that people who ought to know better have been claiming all along, people who forgot or never knew that actually trade-offs in everything all the time.
Right, and it's one of the things that I was responding to more than 20 years ago was the fact that people were telling a biological story that made no sense. And the reason that story made no sense, literally never made any sense, is that if it were true that the more telomere you had, the longer you live, then selection would have a very easy time lengthening telomeres. And so there was something implied, the limited length of telomeres. In fact, that limit evolved second. Our eukaryotic ancestors that were single-celled or colonial, like yeast, have telomeres that maintain their length. So that something in becoming a complex organism resulted in the evolution of limited length of telomeres that shrink with time or with cell divisions. And so it was always implied by the facts that there was some reason to have the telomeres shorten. All right, the question was, what was that reason? So let me read her second paragraph. She says, the clocks come in the form of caps on the ends of chromosome, the long twisted strings of DNA carrying the cell's genes. The caps on chromosomes, called telomeres, are chains of short repeated segments of DNA. Every time a cell divides, its telomere gets a little shorter, until finally they get so short that the cell dies. Well, that's not true. Okay, that's just simply an incorrect description of what happens. It is true that cells, somatic cells that are not in a small number of special classes, do shorten with every cell division. But it is not true that when the telomeres get to a critical level of shortness that the cell dies. What happens actually is that the cells go into a different phase. And the paper that Debbie Cizik and I published argued that the interpretation that the field had thrown onto this second phase was incorrect. We argued that it was in fact an adaptive response to having reached the end of their capacity to reproduce. And the upshot of the paper was that the reason to have that limit was that it regulated the runaway cells where the repair capacity had been triggered to go into a phase that was unlimited and they would otherwise become cancers were it not for the fact that the telomeres shorten with each cell division and rein them in. We would be overrun by tumors.
Runaway cells being informal speak for cancer.
Well, runaway cells being informal speak for tumors, and then tumors become cancers when those cells start metastasizing and spreading around the body. But nonetheless, yes, it was tumor suppression versus tissue repair, telomeres being the regulator that balances these things, and the balance being the reason that we have such great longevity, right? You don't want long telomeres or short telomeres. You want balanced telomeres because they provide the optimal balance point between the two hazards. So what I'm getting at here is, it's very interesting. This New York Times report, again, reports on a New England Journal of Medicine paper, one that the New England Journal of Medicine thought important enough that not only did they publish the paper, but they published an editorial on it because the significance is so substantial. But what is so very odd is that this is reported as if nobody saw it coming, when literally my co-author and I published a paper more than 20 years ago. Yeah, let's show that paper. Yeah, you want to put it up?
Screen?
No, I got it.
Oh, you have it.
Show you a screen.
So here it is. This is, and I should point out that you will see this is published in the Journal of Experimental Gerontology. The title of this paper is The Reserved Capacity Hypothesis, Evolutionary Origins and Modern Implications of the Trade-off between Tumor Suppression and Tissue Repair. Now, this paper has a bit of a history to it. This is actually a publication in Experimental Gerontology following the rejection of a paper that was very similar to this by Nature, one of the two premier journals in the world. Nature preposterously claiming that the topic was not of sufficiently broad interest to their audience to be worth sending out for review. Nature actually officially refused to review the paper, in spite of the fact that the paper was sent to them. Yes, Debbie Cizik and I were both graduate students at the time, and they could have overlooked the significance of the paper, but it came to them with recommendations from George Williams, one of the greatest evolutionary biologists of the 20th century, who specifically recommended to them that they take it very seriously and review it, and a similar letter from Dick Alexander, my and Debbie's PhD advisor, a member of your committee, who also strongly recommended that they take it seriously.
And both, I presume Williams was, but both members of the National Academy.
Right. These were, you couldn't get a stronger recommendation for an evolutionary hypothesis than those two, and yet Nature flatly refused to review it. They sent it back saying it just wasn't sufficiently interesting. So after that happened, something that I have to speculate a little bit about occurred, which is that I got a letter from Experimental Gerontology saying that they had heard about the manuscript and that they were interested in us submitting it to them, which we did. I believe that that happened because George Williams must have contacted them back channel and told them that it was worth their time. In any case, they ended up publishing it finally in 2002. And in any case, it has been not widely discussed in cellular and molecular biology. And as you can see, it takes something like 20 years. And finally, cellular and molecular biology is catching up to the idea that there is a fundamental trade-off that is adaptive in nature. And there is enough of a hubbub about it that the New York Times is reporting on it. Now let's look at the paper that the New England Journal of Medicine published. So not the editorial, but the primary paper. Yeah, the original article. Okay. So the title here is Familial Clonal Hematopoiesis in Long Telomere Syndrome. And what this paper describes, so there's a lot of inside baseball here. There are various things that were predicted in my original paper that turn out to be true. Among them, the idea that moles are actually cells that have run up against their telomeric limits. We called them prototumors, which I still think is a great term that ought to be adopted, but I've never seen it used by anyone else.
Dermatological moles, not Avogadro's number moles.
Not Avogadro's number or,
moles.
Right, exactly. So I won't go into all of the detail, but what they have done here is they have done the flip side of another piece of work. So back when we originally published this paper, there was a question about whether or not progeria syndromes, that was accelerated aging syndromes, the worst of them being Hutchison-Gilford's progeria. People have probably seen images of these usually boys, these very young boys who appear to be very, very old.
They look like wizened old men.
They look like
they're patently 6, 8, 10 years old.
Yes, and they have virtually every pathology that comes with old age except for two. One of them is cancer and the other one is cognitive decline. So interesting that those two don't show up. That would be predicted by our hypothesis. But in any case, the reserve capacity hypothesis did not exactly predict what was going on with Hutchison-Gilford progeria. It turns out there was a second mechanism we didn't think of, but we predicted that Hutchison-Gilford progeria and other progerias would be the result of short telomeres. It turned out that people who had these disorders had normal length telomeres that shortened at an abnormally fast rate as a result of the fact that cells were produced but didn't stick. So if you had to produce 5 cells to get one that stuck, it resulted in a 5 times the rate of expenditure of telomeres. So, okay, if you have shortening telomeres that accelerate or accelerated in their telomere shortening, and it gives you pathologies of aging except cancer and cognitive decline, which presumably functions through an entirely different mechanism. What happens if you have very long telomeres? Well, that's what this is about. So they studied a syndrome called, can you scroll down to the abstract here? They studied a syndrome called...
Is it humans that they're looking
CHIP. Yes, they studied humans. It was no, back up. Okay. So they studied people who have a germline mutation in a gene called POT1. Now, normal POT1 is involved in the shortening mechanism of telomeres that causes telomeres to erode with cellular proliferation. Mutations in this gene result in either additional telomere being added or a reduction in the rate of shortening. But either way, it results in somatic tissues out in the body, that have abnormally long telomeres. And lo and behold, what did they find? But they found that people who have this mutation were disproportionately prone to certain types of cancers and a couple other pathologies.
Quote, a range of benign and malignant neoplasms involving epithelial, mesenchymal, and neuronal tissues in addition to B and T cell lymphoma and myeloid cancers.
Right. Okay. Now, there's a lot in there, but the basic point is, the reserve capacity hypothesis turns out to be true. Mm-hmm. Short telomeres shortening telomeres results in increased pathologies of age. Elongated telomeres result in an increase in the chances of developing a tumor or a cancer. So that is fascinating. Mm-hmm. Now, it's strange, though, that not only does the New York Times fail to mention that there was an existing evolutionary hypothesis that predicted exactly this result, but The authors of this paper also do not cite it, nor do the authors of the editorial about the result. They both ignore it completely, even though the editorial, for example, uses a term about the rescuing of cells from senescence by a mechanism that adds telomere, possibly involving the POT1 mechanism, the POT1 gene. So I guess, first of all, I don't want to be in a position of claiming that I know what the significance of this work is. That's not mine to say. We're not supposed to be describing that about our own work. On the other hand, what do you do in a world where you do work, you do it the right way, it is scientifically exactly what we are asked to do, which is to predict things and to demonstrate that what we have put into the world has explanatory power on this basis. Others are supposed to cite it. And what do you do?
Yeah, what do you do? There is a lot wrong with modern science, obviously, as we have spent a lot of time talking about here and elsewhere. The scientific process is often thrown under the bus in favor of accomplishing political ends that actually have nothing to do with science. We have talked a lot about how very non-fundamental and non-central peer review, certainly in its modern instantiation, is to the scientific process. In its modern instantiation, it being imminently gameable and thus gamed almost entirely. But citing those whose ideas you are invoking is absolutely necessary, not to come to know more things, but to keep track of where good ideas are coming from. And this is something that, this was one of the things that I used to insist on my students getting right. And, these are undergraduates everywhere from advanced undergraduates to freshmen who would almost always have the question, as most graduate students do, as frankly many probably practicing scientists and researchers do, and medical researchers do, under what circumstances do I cite? Like why are scientific papers such a damn disaster to get through for people who aren't scientific, because you always got citations in the middle and it impairs the ability to just read through, right? You cite any idea that is not common knowledge. Okay, that is easier said than operationalized, because at what point does something become common knowledge? There will be some disagreement at that categorical border, because the border itself is fuzzy, right? Well, most people agree on this at this point, but not everyone. Do I still cite the original? I would tend to err on the side of giving the credit if it's not really clear, but many people think, everyone, scientist or not, has in their head like plagiarism is bad. Right? Plagiarism is bad because ideas are not in the air. They emerge from individual people's brains, and generally they emerge from the collaboration between people's brains at the point that it's conscious. But we need to keep track of where the ideas are coming from and who's got the good ideas. And very, very often you find there's ways of doing less work, which is a kind of cheating in science, where you say, well, I know this is true, so I'm just not going to bother trying to figure the citation out. Sometimes that's what's happening. You also have the, okay, this paper I'm reading, that's a good review, and so I don't have to go back and read the 700 papers they reviewed. I'm just going to read this review. Oh, they claim this paper written in 2012, this really good review paper published in 2012, and there's no reviews done since then, claims that this work in 1998 found X. Okay, I'm just going to claim that I read that paper, and I'm going to cite that 1998 paper about X. Well, when you actually go back to the earlier paper and look for the claim X, it is alarming.
How often
Alarming how often the claim that was made by the later paper just got it wrong. Yep. Right? And so if you are making a claim based on someone else's claim, the honorable thing to do, the honest thing to do, the only scientifically rigorous and careful thing to do is say, this claim was made by such and such according to this later person. But scientific journals don't want you doing that. They want you finding the original resource. They want you finding who said it. So where in all of this does your and Debbie's paper from 2002, which was based on work that you had been doing since the mid-late 90s, where does that fall into this? Somehow you got disappeared, right? Your work got actively disappeared, and it became totally, not just totally okay, but actually expected, I think, that your work, which is hypothesis-driven, theoretical, specifically makes a number of predictions that have been borne out in the years since. We will not talk about that. And that we will not talk about that thing among scientists, deeply anti-scientific, corrupt, and a problem for all of us, because that way is the end of science.
So this goes to a number of places. You hint at one of them when you talk about hypothesis-driven. Proper science is hypothesis-driven. Mm-hmm. Most people think that it's clever to say that their science is data-driven. As you and I have talked about, data-driven is actually a kind of a coup that has been mounted against hypothesis-driven science by laboratory types who have various reasons, perverse incentives for not wanting to be so closely obligated to the hypothetical realm. But this is a case. For one thing, evolutionary biology as a field has a right to be recognized when it succeeds in predicting things in the laboratory. In other words, is this just is it an abstraction? Is it like, studying distant quasars that it has no meaningful application on planet Earth? No, this actually has important medical implications and important funding implications. You have entire fields that have been studying what is obviously a dead end from the point of view of dealing with a pathology. They've been lying to each other about what is likely to work. Are we likely to extend life by increasing the length of our telomeres? Are we likely to cure cancer by getting our telomeres to be shorter, right? Those two things have to come together and be discussed, and the point is you need a proper theoretical context. How do you know what a proper theoretical context is? It makes predictions. How good are the predictions? I don't know. How much realm are they relevant to, and how early do they come?
This is 20 years. And how risky were they? It sounds counterintuitive, right? But you want risky predictions, because a non-risky prediction, that couldn't possibly not be true, isn't much of a prediction. If the thing is almost certainly true under a whole array of circumstances, it's not much of a prediction. A risky prediction is true if and only if the hypothesis from which it comes is itself true.
Right. And there's a question about the, so the work that it turns out 20-some years later was predictive of this result that both the New York Times and the editors of the New England Journal of Medicine apparently think is very important, even if readers of Nature wouldn't have thought it was worth their time back in 1999. But the implications may extend far beyond those predictions. In other words, we don't predict things in order to see the results of an experiment early. We use experiments to figure out which models of the universe are actually closely aligned with an underlying reality that we can't observe directly.